자, 이제 now it's time to 10:30, so we will start our class. Okay, just open the INU Smart Campus apps to check your attendance and just enter 9733. To check your attendance, just enter 9733. I will check your attendance. Okay.
Recording.
Okay. And also open your cams. 조영훈, 이상건, 노유미. Just open your cam. And just also enter 9733 in your INU Smart Campus app to check your attendance. Okay. Okay, just open your cam. I just want to see your face. Okay, just open your cam, Isuyeon, Leia, Lonika. Okay. Okay. Okay, just enter 9733. Okay, so now we will start our class. Okay. Okay, just open your cam. Okay. Okay. So, okay. Last time I announced our average or midterm exam. Okay. So now our midterm exam, the average is about 95, maybe, and also 10% get the full score. Okay, so if you got there, if you score over 93 or something like that, you will have higher chance to get A+. Okay. Okay. And also we have, I will announce again the final term exam. In the final term exam will be held. Okay, Lonika, why?
Sorry, Professor, I was just to inform you that my internet connection is unstable. Is it okay if I cut my video? Because you're buffering.
Lonika, once again?
I said my internet connection is unstable. So is it okay if I cut my video?
Okay, it's okay, it's okay, it's okay. It's okay, okay, don't worry, okay, okay. So our final term exam will be held on December 10th, from the 10:30 to unlimited. And I think all of you will also solve the final term exam within 1 hour. However, I will give you our exam time unlimited. So take your time, okay? Just test range is from the chapter 6 to chapter 10, and test is full score is 50, and test method is you have to connect to Joon. Okay, and after you have to solve the test question while showing yourself in the real time through Zooms, like the midterm exam. Okay. So before we start our final term exam, I will check your status, all of your status, okay? If you are okay, you are not okay, you are okay, you are not okay, okay? Okay, so and also you can download their file from the 15th week assignment at from the 10:30. Okay, so you can print the file, or if you don't have print at your home, just write down your answer in the A4 paper. And the important thing is this one. So after you finish final term exam, you have to write your name and your name finished the exam in Zoom chatting window. Okay, in the Zoom chatting window, you can say that finish the exam like this. So I will check this one. So I will check your time. At that, you have to take a picture, your answer sheet. Okay, and Marin? Okay, do you have any question? Okay, so take a picture, your answer sheet, and also send it to my phone, or you can send it to my KakaoTalk within 5 minutes. Within 5 minutes, okay? So I will also check this time. After that, make an answer sheet as PDF version, okay? And also submit your answer sheet through assignment in e-learning, okay? So do you have any question in the final term exam? Any question? Okay, so okay, some of them you are right. So you can enter 9733, okay, in the INU Smart Campus apps, okay, to check your attendance, okay. Okay, do you have any other question? 문경록.
네.
카메라 안 켜?
아, 저 카메라 켰습니다. 교수님. 휴대폰.
오케이. 이윤성. Okay. Okay, just open your cam. Okay.
예. 켜져 있습니다.
Okay. Okay. And also our question languages for final term exam will be given in both Korean and English. And you can write down your answer in Korean or English, okay? Also, this is the detailed information of final term exam. The total score is the 100 points. So homework, you already got 50 points. So final term exam is 50 points. Okay, so from the chapter 6 and chapter 10. Okay, we have the multiple choice, and also we have you have to write down the answer. Okay, this is the problem number and this is point. Okay, like this. Okay. And also, okay, this is homework submission status. Okay, 한동혁, you have to submit your chapter 10 homework. You didn't submit chapter 10 homework. Okay. And I will also give you another assignment to this one. Okay, pre-check task for taking the final term exam. I found that 2 persons did not submit their video. Okay, I'll check. So I found that who did not. Okay, 이도영. And who did not submit? 천준영. So you did not submit the video file. So it means that you will lose your chance to take the final term exam. 천준영 아까 보이던데?
모르겠습니다.
왜 안 냈어?
이거 안 올라간 것 같습니다. 올렸는데.
다시 내 알겠지?
네, 알겠습니다.
If you did not submit your the video, it means that you are not ready for final term exam. It means that you will lose your chance to take the final term exam. Okay. 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 Lee Do-young is also, he will lose his chance to take final term exam. Okay. Okay. So today we will cover from chapter 6 to chapter 7 and part of chapter 8. Okay. So now we will start our chapter 6. So, okay, so do you have any question in chapter 6, class material or the homework? Do you have any question? Do you have any other question? Okay, it means everybody is understand the chapter 6. Okay, so we will see. So chapter 6 is covers the mono-allelic expression. So mono-allelic expression is composed of two subjects. First one is genomic imprinting. Second one is X inactivation. Okay. So this is one example. So biallelic means that both allele, okay, for example, large A allele or small A allele, both alleles can be expressed. However, the mono-allelic expression is only one allele can be expressed. See, this is due to the just imprinting or the X inactivation. Okay, so this is the one example. Is the genomic imprinting is the parthenogenesis. In Korean, we call it as 전혀생식. So it means that reproduction without fertilization. Okay, so allele inherited from the father selectively can be expressed. Or another case is allele inherited from the mother is selectively expressed is like this. Okay. Okay, and also another important thing is that we can see the example of genomic imprinting in the IGF2 genes. It's very important, okay? Okay, so IGF2 gene is the full name is the insulin-like growth factor. So this IGF2 gene plays important role growth and development of the fetus. However, paternal allele of IGF2 is actively expressed in the fetus, but the maternal from mother allele is completely silent in the fetus. Okay, so now we will see some detailed information. So this is the maternal allele of IGF2. Okay, to solve the final term exam, you have to understand this mechanism. Okay, and also we cover our homework. We cover that some detailed information in the multiple choices like this. So DNA methylation occurs. So the right one is the DNA methylation occur in the ICR region, means that which prevent CTCF from binding, so resulting in the expression of the IGF2 by enhancer like this. So okay, let me see like this. Okay. So this is the maternal allele. So okay, there is no methylation in the ICR region, imprinting control region. So ICR region contains methylation sensitive CTCF binding sites. So there is no methylation. It means that CTCF can strongly bind to the ICR. After binding, H19 gene is activated. However, so to express the IGF-2, it needs enhancer activity. But IGF enhancer is located around this region. So CTCF, so we see there's some example of how does CTCF works. So CTCF blocks the interaction between the enhancer and the IGF-2 promoter. It means that maternal allele of IGF-2 is completely silent. Okay, you have to understand this mechanism. Okay. Okay. And also we cover in our so how about this one? Okay. And also paternal allele. Okay, in paternal allele, and there is methylation in the ICR region. It means that CTCF cannot bind to the ICR region. Okay, it means that there will be the interaction between the enhancer and the IGF-2 promoter. So it means that paternal allele over IGF-2 becomes active in the fetus. Okay, so this is paternal allele. Okay, and also we learned that this one is also very important. It's very important you have to understand this concept to solve the final term exam. Okay. There is a two case. For example, one case is that paternal allele of IGF-2 is normal. However, the maternal allele of IGF-2 is mutant. So in these cases, the mouse should exhibit normal size mouse. Another case, paternal allele of IGF-2, they are mutant form. However, maternal allele of IGF-2 is they are normal. So in these cases, in this case, even though the maternal allele of IGF-2 they are normal, but they cannot be expressed. So therefore their phenotype with dwarf, they will exhibit very short status like this. They are very small like this. Okay, so we call it as a dwarf mouse. Okay, so you have to understand this concept. And also so in our homework, we cover this one, okay? So in these cases, so you have to understand to solve the homework 11, you have to understand this concept, okay? Now, finally, the percentage of the percentage will be the 50% like this, okay? Okay, this is very important, okay? Okay, so another case is of the genomic imprinting is that AS gene and PW gene is also another example of genomic imprinting. Okay, and also this is very important, okay? So you have to understand this concept to solve the final term exam. Okay, for example, AS gene, the full name of the AS gene is Angelman Syndrome gene. So this gene plays an important role in the nervous system. Okay, so they are located in chromosome 15. Okay, so however, the paternal allele of AS gene is completely silent. Okay, but the maternal allele of AS gene, they are actively expressed. Okay, so But when the Angelman syndrome is caused by when the maternal allele of AS gene is deleted or mutated. So this is the one case. Okay, another case is PW gene. So PW gene, we call it as the full name is Prader-Willi syndrome or Willi syndrome genes. They are very important role in the normal development. So if they have the Prader-Willi syndrome, they will show that very obesity or small hands and feet and short status, and also they will exhibit mental retardation. Okay, and in this case, PW gene is totally different from the AS gene. So in this case, paternal allele of PW gene is actively expressed. However, maternal allele of PW gene is completely silent. That's why Prader-Willi syndrome is caused by the paternal allele of PW gene is deleted or mutated. Okay. So in this case, this is one case in Angelman syndrome. Now we will see another case in the Prader-Willi syndrome. Okay. So you have to understand this concept, these two concepts, to solve the problem. Okay. Okay, let me see one example. So one case in the Angelman syndrome. Okay, sperm has the normal chromosome 15. However, the egg has the chromosome 15 in which PW and the AS gene is completely deleted like this, okay? But after fertilization, AS gene is completely silent due to the genomic imprinting. However, the paternal allele of PW gene is actively expressed. That's why they will induce the Angelman syndrome. However, they cannot induce Prader-Willi syndrome. This is one case. Another case is that in this case, egg, maternal allele, they are normal chromosome 15, but the sperm has the chromosome 15 in which PW and the AS gene are deleted. So after fertilization, so maternal allele of PW is completely silent. However, maternal allele of AS gene is actively expressed. That's why they can induce the Prader-Willi syndrome, but they will not induce Angelman syndrome. It's like this. So you have to understand this concept, and also you have to understand this concept, okay? Very important, okay? Okay. So now we will go to the X inactivation. So as I mentioned earlier, we will cover two subjects. One subject is genomic imprinting. Okay, we have to know the IGF2, and also we have to know the Angelman syndrome. We have to know PW gene. It's okay. Okay, so X inactivation, we already covered the X inactivation in the chapter 4. Okay. For example, in the murmur, we have in mammalian, we have the X inactivation. So random inactivation of one of the XX in the females. So especially, so at chapter 4, during the chapter 4, we did not cover cis non-coding RNA. But now we will cover XCI, X chromosome inactivation mechanism. Okay, so this mechanism is due to the cis non-coding RNA. Okay, so we will see that there is the X inactivation center is composed of the like this. So they are composed of the 5 non-coding RNAs. Okay, but however, the 2 non-coding RNA is mainly expressed. This one we call it as cis, and this one we call it as 6. Cis and 6 are a little bit different, a little bit similar, but cis and 6 like this. But so inactive X chromosome or active X chromosome is depending on the presence or absence of cis non-coding RNA. It's like this. This is cis non-coding RNA. Okay. Okay, so the function of cis is they are non-coding RNA. They can do their main function is a random inactivation of the one of X chromosome. And the function of 6 is that this is totally different from the cis, this one. Okay, this is the antisense RNA of cis non-coding RNA. So this 6 induces transcriptional inactivation of cis. They are totally different function. Okay, they are totally different function. Okay. Okay, so there are 3 models of XCI mechanism. So one is the sensing and the choice model. So you have to understand that this model. This is the most trusted model. Okay, so you have to explain this model. Okay, important. Okay, and another 2 model is that blocking factor model, and also there is the 2-factor model is like this. So for example, blocking factor model, there is no involvement of cis, and also 2-factor model is there is also no involvement of cis. Okay, so we in our chapter 8 homework we also cover that. Chapter 7 homework, we also cover that like this. Now we go to chapter 6. Okay, so chapter 6 we cover IGF-2 this one, and also cis. In our homework we also cover X inactivation in the fruit-wise and human. And also choose an XCI model that explained the factor from autosomal and sex chromosome are complex together. In these cases, we call it as 2-factor. One comes from autosomal, one comes from sex chromosome. Okay, but there are no involvement of the cis non-coding RNA. Okay, so we covered this concept. Okay. Okay, so now I will explain the sensing and choice model. So you have to explain this concept to solve the final term exam. So this is the most trusted model. Okay, and also cis non-coding RNA is involved in this process. So during the early embryonic state, cells start to differentiate. At that time, the 2 X pairing region, they are pairs each other. After that, this pairing is upregulated cis on both X chromosome. both X chromosome is like this. Okay, so and also at that 2 6 2 6 6 6 the 6 so the 6 region, they are parallel to each other. They are parallel to each other. Next one is we have to choose which X chromosome to activate, or they have to choose which X chromosome to inactive. So in this case, one case, if they choose this X chromosome to activate, in this case, 6 is repressed cis expression on X chromosome. In this case, is X chromosome remains active. Another case is 6 does not repress the cis expression on X chromosome. So in this case, X chromosome remains inactive. Okay, so this is X inactivation mechanism. This is most trusted model. Okay, so you have to understand this concept. Okay. Another concept is there is also blocking factor model. There is no involvement of cyst, and also there is the 2-factor model. One comes from the autosome, the other comes from the X chromosome, like this. Okay, so you have to understand this concept. Okay. Okay, and also we learned that initiation of X chromosome inactivation. Okay, so PCR2, especially this protein is bind to the lipid A motif, and also PCR2, another EZH2 domain, they can do methylation is like this. And also we cover transcriptional inactivation and transcriptional activation by the so we learned the transcriptional regulation of cis non-coding RNA in active X chromosome. Okay, so in this case is this one DEX activate the 6, and the 6 induces transcriptional activation of the cyst. And another case is in the opposite. Okay, so we also cover the maintenance of X chromosome inactivation. There is 2 factors involved in the maintenance of the X chromosome inactivation. So first one is the DNMT. So this is DNA methyl transferase. So it will induce DNA methylation. And another one is the macro H2A. So this is the nucleosome factor. Okay, so this macro H2A is incorporated nucleosome of inactive X chromosome. So this one, the main function is interferes the binding or the transcription factor or SNF, the nucleosome remodeling factors to binding. Okay. And also we cover the spreading of X chromosome inactivation. So we call it as a proximity transfer. The reason why is the cyst forms the chromatin loop like this. So cyst, they will form loop is like this. So they will induce special proximity. So it means that it can induce spreading. It can induce spreading of the X chromosome inactivation. Okay. So there is a the mechanism is like this. Okay. Okay, and also we cover X chromosome reactivation is like this. Okay, okay. Do you have any other question in chapter 6? So we finished chapter 6 summary. Any other question? Okay. Okay, so now we will go to the chapter 7. Okay, now we will have the one minute break because I need water. Okay, 1 minute breaks. Okay. Okay. Hey, Zolonica. Is there okay, Lonica is not here. Tibor.
Yes.
Yeah. Is there any other problem in our epigenetics class? Are you okay?
Yeah, I'm okay. Thank you.
Okay, so maybe this subject, this epigenetics is very difficult to understand, especially chapter 8, chapter 9, chapter 10. They are very difficult to understand. Okay. Okay, so maybe tomorrow I will explain some details in chapter 9, chapter 10 is very difficult to understand. So I will explain it tomorrow, okay? Okay. Zana, are you okay? Is there any other problem?
No, it's okay.
That's okay, okay. So I think that everybody's have a higher chance to get A+ if you got a good score in the final term exam. So maybe most of students will get good score, I think. Okay. Okay, and last year nearly 90% of students got A+. Okay, I don't know this year, but okay. 신주환,
네, 교수님.
수업 잘 듣고 있어?
네, 열심히 따라가고 있습니다.
세무 회계학과 힘들 텐데.
네, 쉽지는 확실히 않는 것 같습니다.
많지. Okay. Is Marin? Marin?
I'm fine for now.
You are fine. Okay. So please understand actually the epigenetics course is very difficult, but I try to explain it very simply or very detailedly. Okay. Okay. 한동혁 이 수업도 안 듣고 있고 숙제도 안 내고 있고 이거 한동혁. Okay. Anyway, okay. Okay, so now we will cover chapter 7. So do you have any other question in chapter 7 in the class material or homework? Do you have any other question? Okay, So now we will cover chapter 7, okay? So RDDM, we call it as the RNA-directed DNA methylation. So this is the epigenetic process first discovered in the plant. So this is initially the host defense mechanism against the virus and the oral transposon in the plant. So this RDDM is usually found in the heterochromatin region, but this is not found in around the centromere region. So this is so transposon is they have the function is like this. So who is studying the transposon? So in my lab, I use the transposon system to actively express the protein. So now our lab, we studied transposon mediated transgenesis in the mammalian cells. So if you are interested in transposon, so I will teach you. I will teach you some specific experiment in our lab. Okay. Anyway, the RDDM is mainly discovered in the plant. Okay. Okay, so there is so RDDM needs 2 RNA polymerase, especially they need, usually during the transcription, usually we know that RNA polymerase 2 is involved in the transcription. But in addition to the RNA polymerase 2, especially the RDDM, we need another RNA polymerase 4 or polymerase 5. It's okay. And also we learned that DNA methylation in plant, so they also occurs in CHG or CHH region, where H is A or T or C is like this. Okay, and we covered MET1 and CMT2 and 3, and also we covered DRM2. So DRM2 is highly related to RDDM. This is the de novo DNA methyltransferase. So de novo DNA methyltransferase. So this is nearly similar to the DNMT3A or 3B. It's like this, okay? This is very important, okay? Now we will go to the canonical RDDM, and also we will go to the atypical RDDM, okay? And because the chapter 7 is the main subject of chapter 7 is RDDM, okay? Especially the canonical RDDM is 99% of cases is the canonical RDDM. Okay, only 1% is the atypical RDDM. So we have to know this canonical RDDM to solve the final term exam. Okay. Okay. Especially the RNA polymerase 4, this one, they can bind to the methylated region, okay? To initiate the transcription, usually the RNA polymerase 4 have to bind to this region, DNA region. But in this region, the DNA is methylated. However, RNA polymerase 4, they can bind to the methylated region. Okay, so RNA polymerase 4, they can synthesize RNA transcript. Okay, after that, RDR2, they synthesize double-stranded RNA. After that, DCL3, they can cleave double-stranded RNA into siRNA. Okay, this is step 1. And also at that hand 1, they induce this. We already learned the hand 1 in previous chapter 4. 3' methylation. At that argo 4, they bind to the double-stranded siRNA and cleaves it single-stranded siRNA. They will make this argo 4 single-stranded siRNA complex. So this is step 1. How about step 2? Step 2 is, in this case, is RNA polymerase 5 is involved in the step 2. So RNA polymerase 4 is bind to the DNA region. They can synthesize RNA transcript with like this. So in this case, so at that previously they made this complex. So this complex is bind to the nascent. Newly nascent means a newly synthesized RNA from RNA polymerase 5. So it will recruit DRM2. So DRM2, they can induce de novo methylation. This, okay. Okay, now we will go to the step 3. We call it as a chromatin alteration. So this is we cover step 2, and they will make the nascent RNA. Nascent means a newly synthesized RNA. So usually the IDN2 and IDP complex bind to the RNA transcript, and also they bind to the 3-SNF complex. So this binding is inhibit 3-SNF complex activity. It means that the main function of 3-SNF complex, they will lose the chromatin structure. But it means that this binding prevents the step to loosen the chromatin structure. And also another SUVH4 or 5 or 6 is recognize the methylation and decode information, and also the HDAC 6 is the removes histone acetylation. It means that they will induce packing of chromatin structure. It's like this. Okay, very complicated, but you don't have to memorize the component name. So I will give you the so this is the full picture of the canonical RDDM. So I will give you the full picture like this. So you have to explain the procedure, okay? They are composed of the 3 steps. First one is the RNA polymerase 4 dependent siRNA biosynthesis. Second step is that we call it as the polymerase 5 mediated de novo methylation. Due to by the DRM2, they will make the de novo methylation. And finally, there is the chromatin alteration step. Okay, so this is the canonical RDDM mechanism. So you have to explain this full procedure, okay? Because this is the main subject of the chapter 7 is RDDM. Okay, so you have to understand canonical RDDM, okay? And also there is another step is atypical RDDM is composed of step 1 and also step 2 and step 3, like this. Okay. Okay, let me start from the step 1. In step 1 is usually during the RNA transcription, RNA polymerase 2 is bind to the DNA. So in this case, there is no methylation here. In this case, RNA polymerase 2 is bind to the DNA. They can synthesize the RNA transcript. So in our chapter 7 homework, we covered this one. We covered that describe how polymerase 4 contribute to the spreading of the DNA methylation. Okay, so usually the RNA polymerase 4 bind to the methylate the DNA region. But in this case, in the RNA polymerase 2 cannot bind to the DNA methylated region, but RNA polymerase 4 they can bind to the DNA methylated region. Except that RNA polymerase 4 creates a transcript, synthesized transcript to generate siRNA. After that, they will allow the spreading of the DNA methylation. Okay, spreading of DNA methylation through the process of canonical Rddm process. Okay, so we cover this one. Okay, and also, so there is no methylation. In this case, RNA polymerase 2 is bind to the RNA transcript. And after that, RDR6 is they synthesize double-stranded RNA is like this. Okay, and they will make the complex is like this. Okay, and second step is that polymerase 2 dependent RDDM. Okay, so usually the DRM2, they made the de novo methylation, and it means that they will establish transcriptional gene silencing. So if there is the methylation here, so usually the RNA polymerase 2 cannot bind to the methylated DNA. So it means that they will induce transcriptional gene silencing. Okay. And also step 3 is that we call it as a polymerase 4 and RDR2 dependent RDDM. Okay, so they will make DCL3, they will make secondary siRNA. And finally, so this step. So finally, the polymerase 5 is synthesized is the RNA transcriptor. At that, previously they made the secondary siRNA and they made complexes like this. At times, RNA single-stranded siRNA complex is bind to the nascent RNA from polymerase 5. So it will recruit DRM2. So DRM2 induced de novo methylation in the neighboring region. So resulting in DNA methylation spreading and the reinforced transcriptional gene silencing. So we cover this one in our homework. So we explained how secondary RNA can spread the gene silencing to the surrounding region. Okay. So, for example, this in atypical RDDM, DCL3, they can make the secondary siRNA. After ARG4 bind to the secondary siRNA. After that cleaves in single-stranded siRNA, and finally they will make the ARG4 single-stranded siRNA. So it means that this complex is bind to the RNA transcriptor. After that, they will recruit DRM2. So they induced de novo methylation in the neighboring region. So it will induce DNA methylation spreading. Okay, so it will reinforce transcriptional silencing. Okay. Okay, so initial step there is they made the DRM2 made some methylation here, but secondary siRNA they made another methylation around here like this. So this one we call it as DNA methylation spreading. Okay. So we cover in chapter 7 in our homework, we cover this one, okay? Okay, so targeting polymerase 4 to the chromatin. So in this case is the two proteins. For example, first one is SHH1 and the CLSY1. So this one is the first one, SHH1 recognize the methylation. And after that, CLSY1, they lose the chromatin structure. After that, RNA polymerase can bind, RNA polymerase 4 can bind to the methylated DNA region. Okay, so we cover this one, these two concepts in our multiple choice. So multiple choice is choose which of the following paired correctly as important for chromatin specific binding or polymerase 4. Okay, two protein is involved in this step. First one is SHH1. This is bind to the methylated region. And after that, CLSI1 is they do lose the chromatin structure. Okay, and also we cover the for example, especially polymerase 5. Okay, in this case is polymerase 5. In this case is DDR complex. DDR complex is promote targeting of polymerase 5 to the chromatin. Okay, so we also cover this one in our homework. Okay, for example, consists of the DRD1, TMS1, RDM1, and it promotes binding of the polymerase 5 chromatin. Okay, this is we call it as a DDR complex. It's like this. Okay. And also targeting of RNA polymerase 5 to the chromatin. So in this case is so in the canonical RDDM. So usually in the second step we call it as polymerase 5 mediated de novo methylation. So at that time, so SUVH29, this one, okay, this one, they bind to the methylated DNA. So at that, they will recruit, they will promote the targeting of RNA polymerase 5 to chromatin, because they have the SRA domains. Okay, so we also cover in our homework, explain how SUGVH2 and 9 is involved in the chromatin specific binding of polymerase 5, because they have the SRA domain. Okay, so this complex is bind to the methylated region through the SRA domain. So this binding is facilitated RNA polymerase 5 to specially bind to the methylated region like this. Okay. Okay. And finally, we cover the Pharah mutation. Okay, very important. Okay, very important. Okay, Pharah mutation is the non-Mendelian inheritance. In Pharah mutation, there is no dominant or no recessive. So during the Pharah mutation, interaction happens between two alleles, whereby one allele induced inheritable changes in the other allele. The inheritable changes include DNA methylation or histone modification. Okay, let me see in these cases. So, okay, so in the final term exam, you have to understand this concept. Okay. For example, there is two allele. First allele is a large R, deci small r allele. This is the enclosed dark purple seed is like this. And another allele is RST allele. So they will enclose the purple spotted seed is like this. And small r allele is they don't enclose no color seed. And the large R-R is dominant than small r. And the large R-ST, this is dominant than small r. However, there is no dominant or no recessive relationship between the RR and RST alleles. Okay, for example, in these cases, they cross each other, so they will make the offsprings should be like this. Like this. This one is a large R dash small r slash small r. In this cases, this one is dominant than this one. Their phenotype will be dark purple seed. It's like this. And in these cases, this one is dominant than this one. So they will enclose purple spotted seed is like this. However, RST allele is recorded as a paramutagenic allele, and RR allele recorded as a paramutable allele. So this paramutagenic allele can induce gene silencing in the another allele. So if that paramutable allele if that RR allele becomes R-R' allele, so this one also become the paramutagenic allele is like this. Okay, so in these cases, okay, so in these cases, large R, small r, and RST allele, because this large R, small r, they will encode the purple seed, and also large RST, they will encode the spotted allele, purple, but the RST allele, because they are coexisted, but we cannot differentiate it. Okay, because R allele, they show that the purple seed is like this. But in this case, RST allele, they Pharah mutagenic. Okay, so they will make the R allele to R prime allele is like this. So in these cases, one becomes the R small r, but this one will become the R small r prime allele, and another allele will be the RST small r allele. This one is this one. So large R small r prime allele encodes lightly purple spotted seed. Okay, because this one is the dominant than the small r allele. That's why they will encode, their phenotype will be the lightly purple spotted seed. And this one is still the purple spotted seed is like this. Okay, so you have to understand this concept. Now also our homework, we have the one example of the Pharah mutation here like this. Okay, so expect the phenotype of offspring in the crosses and explain the region. Okay, so and explain the phenotype of the offspring and crosses and explain the region. Okay, so you have to explain the region is like you have to show the phenotype, and also you have to explain the region, because the large R small r allele becomes the large R small r prime allele. So this one is encoded lightly purple spotted seed is like this. Okay. Okay, so do you have any other question in chapter 7? So we cover very important thing. Okay. So we have to understand the full procedure of canonical Rddm. Okay. And also there is a special, because the RNA polymerase 5, they can bind to the methylated region. Okay. Okay, and also we have to okay, and also a typical Rddm, they will make the secondary siRNA. They will make secondary siRNA. That finally, DRM2 is the de novo methylation. They induced de novo methylation in the neighboring region. And also we have to know the Pharah mutation. Okay. Okay, now we will go to the chapter 8. So do you have any other question in chapter 8? Okay, so now we only have 5 minutes, so we will briefly cover chapter 8. Any other question? So chapter 8, which cover histone variants and epigenetics. So there is the several type of histone variants. First one is H2AZ. Another one is H2AX. And third one is we call it as macro H2A. And another one is 3.1 or H3.3. The final one is stamp A. It's like this, okay? So in our chapter 1, we cover histone octamer. So histone is consists of H3, H4, and H2A and H2B proteins like this. They composed of the 4 histone proteins. And also they also need a chaperone. So chaperone, there are several kinds of chaperone is like this. The main function of chaperone is they helps nucleosome assembly. So they make the nucleosome assembly, and also they help nucleosome disassembly. So this chaperone, some of the chaperone help nucleosome assembly, and some of them help nucleosome disassembly. Okay. So there are several kinds of histone variants. They have a unique function, and also various type of histone. They need unique chaperone. Okay. But this one is very important. So in chapter 8, this one is the very important. Okay. In chapter 8, we cover histone variants and epigenetics, and this is common to all of them. So you have to know the nucleosome assembly and disassembly procedure. First one is let me start from the nucleosome assembly. So first one is the H3 and H4, they make a dimer, and H3 and H4 dimer, they assemble, they will make the H3, H4 tetramers like this. So initially start from H3, H4 tetramer. So this tetramer is bind to the DNA. So we call it as this one tetrasome. At that additional two H2A, H2B dimer, one H2A, H2B dimer, one H2A, H2B dimer, they bind to the tetrasome. So they will make the nucleosome. It's like this. So you have to understand this procedure. And also when this assembly happens, so each time they need a chaperone, but you don't have to memorize the chaperone. So just briefly memorize the nucleosome assembly procedure. So another procedure is nucleosome disassembly. So this is nucleosome. Initially, the H2A, H2B dimer, they are lost first. After that, H3, H4 tetramer is lost. After that, DNA is fully exposed like this. Okay, so we have to know the nucleosome assembly and nucleosome disassembly procedure, okay, to solve the final term exam. Okay, so the other part we will cover in the this Friday. Do you have any other question? Okay, do you have any other question? Okay. Okay. So if you don't have any other question, so we will finish our class and we will have another Zoom class and this Friday. Okay. Okay. See you this Friday. Okay. Bye.
Thank you. Have a nice day.
You too.
감사합니다.
Okay. Okay.
교수님,
감사합니다.
저 동혁인데요.
그
아까 전화 받고 있느라 과제 바로 제출했습니다. 했는데 깜빡하고 안 냈어요.
정신 차려. 아, 참.
네.
오케이.
죄송합니다.
오케이.